Julia. So nearly 25% of global population is consuming fecally contaminated water. If you look at this world map here, it is not just happening in the developing countries or third world countries, but it is also happening in the more wealthier countries where we have access to state-of-the-art water technologies. So according to World Bank statistics, this contaminated water is causing about $260 billion annually to the world. So if you look at the, like just in the United States, like about 33 outbreaks were recorded in 2012. So our overarching goal is how can we provide this safe water, which is one of the requirements of United Nations Sustainable Development Goals of 2030. We are trying to develop circular zero economy water technologies, where we take this contaminated water from the rivers or from industrial stream and convert that into low cost adsorption technologies where we can use sponges so that we can produce this gray water applications which do not require advanced wastewater treatment processes. So as a result, we can reduce overall cost and also minimize the wastage and improve the reusability of the materials. So this is where like we are trying to use a sponge filter as a pre-treatment process. So today's talk is how we are designing this nanocomposite sponges using polymeric substrates to remove bacteria from the nutrient-rich water using like adsorption process. This is our research team where like I myself is leading the project from material science and chemical process design aspects. We are also working closely with the microbiology and biochemistry people. Yeah, like Rob is from biochemistry and like Leo is working on the nanochemistry, who's working on the fabrication of materials. Alice is helping us with the synthetic biology of the materials where like we can study like various mutants of the bacteria. And Hugh Williams in life sciences is one of the co-investigators on the project, and Darrell from the chemical engineering is the principal investigator. So if we look at this wastewater, it typically is a mixture of like bacteria, protozoa, viruses, and various other organic contaminations, where we are focusing only on removing bacteria, which is about in micrometer size. So currently there are numerous technologies to deal with this kind of wastewater, from all the way from starting point to the end point, such as like membrane filtration, chlorination, UV treatment, antibiotics. However, these technologies tend to be like very expensive and also time intensive. And sometimes they also become ineffective. For example, one of the quickest ways to kill viruses is using antibiotics, but like because of the antibiotics are used in the water, humans are developing antibiotic resistance to it. So that is another a bigger challenge. So we want to use something like very physical process where we don't have to use like expensive technologies such as UV treatment or chemicals, chlorination or antibiotics. So in terms of like physical process, adsorption is the most cost-effective and simple to use technology. However, this technology is not being fully utilized to its best capacity. That is because like currently available absorbents, such as activated carbon or zeolites, they have like large surface areas, but they also tend to have this nanometer-sized pores. So when we are trying to remove this microbes in micrometer size, these materials are unable to remove them because like these pores get clogged because of the size incompatibility. Alternatively, what we can use is we can use these polymeric sponges, which are like easy to fabricate with the different pore sizes, and they also like are very low cost and exhibit like stable bulk properties. So we can use this kind of material as an alternative adsorbent. And also, but these materials wouldn't have like a best surface properties like the traditional adsorbents. So what we can do that here is like we can make use of the advances, like recent advances in the nanotechnology, where we can functionalize these sponge surfaces for controlled chemistry or topography or charge properties. And we can use also metal-oxide and other nanocoatings, which are proven to be effective in designing antimicrobial surfaces. So what would be the precise properties that we need to be looking at, like when we are designing these sponges? So for that, we need to consider many things, not just the material chemistry, what we should be looking at. So our approach is like, we are designing to actually implement for practical applications. So we need to go through like what are the environmental standards like for a specific target market, and what would be the material cost, whether it is scalable or not. And once again, whether it is commercially viable for the market that we are focusing on. For example, in low or medium income countries, we cannot use like materials that would work very well, but they cannot afford to actually use them when it comes to implementation process. So from day one, we are considering all these factors. And also we are using integrated design approach. That is because like this bacteria, like they have been around for billions of years around on the earth. So they learn to adapt to the environmental conditions. And like depending on the nutrients or water, like whether there is enough water is available or not, temperature or sunlight, for these harsh conditions, they learn to adapt and evolve themselves. So like because of this evolution, their chemical and physical property changes happen. So we need to design surfaces that are also adaptable as the bacteria and still serve the purpose, still serve its functionality as we designed originally for that. In this case, like how we can attach the bacteria to the surface. And also like, despite of having this excellent adaptation capability, it should be also like a low cost, reusable material, and also shouldn't have cytotoxicity effect, which means like it can damage the cells, but it should not damage the human DNA or RNA. And also these materials needs to be scalable. So to understand that, what we need to do is like we need to look into how bacteria behave in the first place and how do they attach to the surfaces. So like there are different intermolecular forces and surface topographies that would help these cells to attach to the surfaces. And if you closely look at this bacterial cell surface, like here, like this organelle has like a different parts to it at the surface. Like there is cell wall, and we have this pili and flagella, curly, like they all have different chemical compositions. Depending on that, they can attach to surfaces via electrostatic attractive forces or van der Waals forces, which are easier to deal with when it comes to like, if you want to selectively capture this bacteria and remove them for further reuse. And we can also use hydrophobic forces if this pili is like the bonding mechanism, like bonding is happening through the pili. And also depending on the topography, we can have this cells attachment to surfaces via steric forces. And we can also bind them to surfaces via chemical modification. But like for us, like because we need to reuse the material, like working with this weaker van der Waals forces or electrostatic forces or hydrophobic interactions would be more useful. So if we look at the like biofilm growth, like at the beginning, the bacteria is suspended in the water, like they are in a free state, like wet swimming state, and they tend to attach to the surfaces where the surface is nutriently rich. Like if we look at the water, like in the water, like for example, we have like these suspended nutrients, but these nutrients tend to settle on the surface because of the gravity. So the surface tend to become like a nutrient rich environment compared to the water, like overall water environment. So the bacteria would tend to like come here in the search of food and tend to form a biofuel. So if it is at the very beginning stage, it would be much easier to like capture them and remove them. but when it comes to a matured biofilms, it is very difficult. So like for now, we are only operating at the stage 1 and 2 in this project. So when we are like trying to select the suitable material for the sponge adsorbents, what we chose to use the polymers. That is because these are the low cost materials and easier to scale up. They also have this stable structures, which means like the polymers are cross-linked structures, which makes them like resistant to harsh chemical, thermal, or like solar environments. So they can still they do not degrade easily. As a result, they can work a long time for wastewater treatment application, which tend to be harsh chemical and physical environment. Similarly, and also like the sponges, their pore size, we can change them anywhere between tens of microns or even nanometer to like hundreds of microns. So there is this flexibility of changing the morphology of the polymers that tend to be only 1 or 2 step process. However, as I mentioned before, they do not have the ideal surfaces, which can be tailored using the nanocoatings, which comes in the next stage, in the next slide. And in terms of selecting the polymers, we do have these natural polymers, which comes from the plants, such as cellulose or chitin-based polymers. Like currently, there is still development is going on with the biomaterials. They are not as stable as the synthetic polymers tend to be, such as polyethylene, polyamide, and polyurethane. So currently we are working with the synthetic polymers because we want a material that is designed not to break. And to tailor the surface properties of the material, what we can do is we can use metal oxides. These metal oxides, such as copper, silver, they also serve as a minerals at the surfaces where the bacteria would want to consume these minerals. So but they tend to consume more than required, which tends to damage the surfaces. So as a result, these metal oxide coatings can also serve as the both a material that can attract the bacteria and also kill them eventually if required. And there are some concerns in using the metal oxides because they also reported to have adverse effect on the human DNA. However, if we are using in the nanomaterial form, we are using only small concentration of the material and still be able to achieve the performance levels as high as we want. For example, if we use a coating, instead of whole the material, we can use only about 1 to 2% of the coating or up to maximum of 5%. So because of this lower concentration, we wouldn't experience that much adverse effect. And here, our approach is using techniques from physics, chemistry, and biology, and actually predominantly materials engineering techniques, where we can apply this techniques such as spectroscopy, X-ray tomography, and light microscopy to study the interaction between the materials and the cells, like the bacteria, and also design the process or evaluate the effectiveness of the process for bacteria attachment and their regrowth. So now I will walk you through the simple method that we have developed, how to design these bacteria for different for control surface properties using nanocoatings. So what we have done here is we have taken the commercial polyurethane sponge, which has a large pore size and a bimodal pore size distribution. But this polyurethane sponge, the specialty about this specific sponge is it has both carboxyl and amine groups. So the advantage of these groups is in response to the pH of the surrounding water, it can switch between positive and negative charges. So we can, this charge property would be beneficial in catching the bacteria from the surrounding water via electrostatic attractive or repelling them from attaching to the surface. However, they do not have this ideal surface energy, which is thermodynamically freely available energy with which the bacteria can also attach to surfaces via van der Waals and hydrophobic forces. So for that reason, what we can do is we can use this nano coatings. These nano coatings can be functionalized with different like alkyl chains or amine groups. And we can also use them in different shapes, like whether particles or wires or pillars, where the spiky structures tend to break the cell membrane much easier compared to the other structures. And they can be in any size, like up to like in tens of nanometers to up to a micron. And like as I said before, like we can use this material as a coating. So an amount of like about 1 to 5% should be enough to achieve required properties. So when it comes to scalability, the manufacturing process needs to be very simple. So like here we developed this simple like two-step process where like we have this commercial sponge which can be dipped into this functionalized nano suspension. Like this can also be obtained commercially, or we can make them in-house. And just we can dip the sponge inside this suspension and remove it and vacuum dry it or bake it to make sure this nano coating is strongly adhered to the surface. And that's all. And we can use it like as much as possible. So like after functionalizing the surface, we need to ensure like what kind of surface chemistry does it has, whether like how many hydrophobic groups, amine and carboxylic groups, where we can use spectroscopy techniques such as X-ray photoelectron spectroscopy, where we can monitor these different functional groups on the surface and at what concentrations they do exist. Also, we can look at the topography of the materials using scanning electron microscopy, where here like what you are seeing is this nanoparticles, spherical nanoparticles deposited on the sponge surface. And we can also measure like what would be quantify the roughness of the nano coatings. That is like this, they provide this multi-scale roughness on the sponge surface, which makes them like very helpful like in binding the bacteria strongly to the surface. And that can be measured using like optical profilometry at microscopic level, at micron level, and atomic force microscopy at a nanometer scale. And in addition to that, like this is a sponge where like we need to know like how the bacteria, like the volume of the bacteria itself is inside the sponge. So this would help us like developing the protocols on like, for example, like on how to reuse this material or controllably kill this bacteria. For example, here, like I'm using this X-ray tomography technique to see like where this volume of the water is inside the sponge. So like if we can also look at the bacteria volume like this, we can target it like instead of using the, for example, UV light, which tend to be very expensive to use for killing the bacteria, instead of using it like uncontrollably, like for like a whole volume of the sponge, we can just target at one place and kill the bacteria there itself and reuse the sponge, which makes it much more energy efficient. Also, like if, for example, if the bacteria growth pattern is something like this, which is spreading horizontally, which means like it is chemically binding to the sponge surface, then we need to develop some other like chemical treatment methods or surface displacement methods to remove these surface biofilms. So next, like we have done some proof of concept experiments where like we did these pristine sponges, like how they would measure up to like removing the bacteria from the nutrient rich water. So as I said, like the sponge has both like this carboxyl and hydroxyl groups. Because of that, depending on the pH of the water, they can switch between either negative and positive charges. For example, for this specific material, at acidic conditions, it is more positive, and at basic conditions, it is more negative. So like if we take a gram-negative bacteria such as E. coli, what happens is like it would attach to surfaces at acidic conditions because of the opposite charge attraction, but it would repel from the surface at neutral and basic pH. As we increase the pH, it would repel further and further. That's what this cartoon here shows. Like at acidic conditions, like there will be a more attachment, but at neutral and basic conditions, the repulsion increases. So here, this is the like gram-negative bacteria, like I used as a model system, which is E. coli, which is very common bacteria that is used in microbiology. And the one that I used is like of micrometer size, because as I mentioned, like we are more interested in using the micrometer size bacteria. Like it is of size like about 1 micron in diameter and 3 microns in length. 1 to 3 microns in length and 0.4 to 0.7 micrometers in diameter. And they have overall net negative charge on their surface. So, and here for adsorption experiments, I chose to use the nutrient-rich water, which is like a LB broth, protein broth. This is because like in the water, like the nutrients are relatively low. So it would be easier to kill the bacteria because they do not have access to like a lot of food. But in the nutrient-rich environment, because they have abundant access to these proteins, the bacteria would tend to like grow and thrive and also form biofilms. So this is a worst case scenario. Like if the material can work for this condition, then it should be able to work for the like much simpler condition, which is like tend to be like a river waste, stream water, or industrial wastewater, like where we don't have the cells do not have access to this much protein, access to the proteins. So here, like the way how I did the experiments is like I took this protein water and mixed it at either at a different RPM, like about 100 to 500 RPM. Then also like maintain the temperature depending on the physiological conditions of the bacteria. Like it can be anywhere at room temperature to 37 degrees Celsius. So like this plot shows the control experiments where there is no sponge, just in this LB medium, which is nutrient-rich water, where like this bacteria this is a growth curve. So over time, like I monitor like how the cells are and like this shows like the cells remain constant. They are not growing, like their concentration is remain the same. And these were like measured at different pH conditions, like acid, neutral, and base. This is to ensure like the pH is uh, not causing the bacteria to die, because like typically they want to like survive around pH 6, like the specific cells. But like if we go like a little bit down to like pH 4 or pH 5, would they survive or die? That's why like we need to do these control experiments. And also to ensure like what is the actual specific role of the sponge. So here, like this specific experiment is ran with the sponge, where like in the LB, like I kept this stationary sponge, so this bacteria can go and attach to the surface. So as I said, like at basic conditions, because these cells tend to repel from the sponge because of same charges. So at basic condition and neutral condition, we see very few cells are attaching to the surface. But when it comes to acidic conditions, like there is most of the bacteria attached to the surface. Like it shows 100%, but it is actually below the detection level of the instrument that I was using. So if you look at like how the bacteria, like it is absent from the water, but where it is actually going to the sponge surface. So to see like where this bacteria is depositing and how much they are depositing, and whether they are forming a biofilms or just attaching on their own at different locations. To verify that, what we can do is we can look under the confocal microscope. Here under acidic conditions, as you can see, like there is like a much bigger colonies of bacteria, like where they are like forming these big aggregates. And but at a neutral conditions, we see very few bacteria at the intersection of the pores. And at basic conditions, like we see even low concentration of bacteria. So this is like obvious, because like they are actually not completely attaching to the sponge surface at a neutral and basic pH conditions. That's why we see very low concentration of bacteria on the pore surfaces at neutral and basic pH conditions. So this is the nutrient-rich water I mentioned, like where this color is actually represents the protein itself. And if the bacteria is in there, it looks more turbid. Like for example, here this case, this is as is like where all the bacteria remain in the water. So it looks very turbid. But at acidic condition, because they are removed, these particles are removed from the protein, it looks more transparent. And again, at neutral and basic pH conditions, like it is a little more turbid. So here I showed only one as is, but actually I did this control experiments at both at acid, neutral, and basic pH conditions, and they all have like a similar high turbidity in them. So once the bacteria is removed, like sometimes like the leftover one, like the small amount, whatever is left over, they can regrow back and become like the turbid environment like this. So what we can do is we can actually, after treating it with sponge, we can take this suspension and expose this water to the nutrient rich surface, here in this case agar. So when we do that, because again they have like access to a lot of food, they tend to regrow. And this is what this tiny dots here is showing, like this each dot is a big colony. And it, as is conditions, we have like maybe like about like more than like 500, like even 1,000 colonies of bacteria that will regrew. But at acidic conditions, I have like only 2 colonies. And at neutral conditions, like about 60 colonies, and basic conditions over 200 colonies. So it shows that it is not the pH, but sponge is helping to remove this bacteria and preventing them to regrow into like a full-fledged uh, uh, big colonies like this. So that is only one species of E. coli, but like depending on the strain of the bacteria and also at what stage they tend to exhibit different conditions, completely different chemistries, different physicochemical properties. So what we need to do is like we need to check whether this substrate would work for other types of species as well. So here I'm testing it with a different strain of E. coli, where to distinguish between the species, like what we can do is like we can actually stain them. This is where genetics play a role, where like we can engineer this bacteria like with the different colors to distinguish between like the species itself and between like the cell, the organelle, cell wall, pili, and curly. We can also distinguish between them so that we can see like actually which one is attaching to the sponge surface. So we can customize the surface chemistry of the sponge according to the uh, uh, interacting surface of the cells. And here this is a staphylococcus epidermis. This is a gram-positive bacteria, and this one attaches to the surface via surface wall, like which is like a alkyl groups. So we also examined these 2 bacteria attachment to the sponge surface. And as you can see here, again, once again, in the control experiments, the concentration of the bacteria remained almost constant, like it only varied between 0 to 20%, which means like it is growing, like where it started at 0, but over time it grew into like 20% of the bacteria actually grew. But when we use the sponge here, these 2 lines, when we actually use the sponge, about 92% of the bacteria went and attached to the sponge surface. And these are only preliminary experiments. In the future, what we will do is like, we will optimize further like the process conditions such as like the mixing speed and temperature to ensure like the bacteria can attach to surfaces at much faster and at much higher efficiencies. And also what we can do is like, we can also engineer the surfaces. All the results I showed so far, it is only for the pristine polymer sponge. So we can have these coatings, different types of coatings on the surfaces, so that as the bacteria evolves and goes through different physicochemical surface properties, surface chemical compositions, we can also have these nano coatings on the sponge surface, which can adapt to these changes in the bacteria and still ensure they attach to the surfaces. And so like we will be further working on like, because it is for real world application, we will look into what are the actual pH in the field, like depending on the geography or the application or the temperature, salinity, and fluid properties itself, like whether it is like a pure water or different water chemistries with different types of organic media. So we can look into those implications as well, how they affect the cells and in turn the design of the sponges. And it would also lead to like, this work would also lead to like different protocols, like adsorption protocols for broad range of process conditions. And based on that, we can also develop this uh, protocols for reusing the sponge via chemical or mechanical cleaning. So all this talks about how to design the sponge, but how about the implementing the sponge? So like here we can actually use a simple system like this, where a column is filled packed with the sponge, and this LB medium can be flown through the sponge to recover, to produce a cleaner water. Like in this case, it shows blue, but like with non-LB water or like low nutrient rich water, then we can have like a more cleaner water. Or in the downstream processes, like such as membrane filtration or direct UV, we can also use those treatment methods to produce water for gray water applications. So like in summary, like actually what we are aiming for is like through this project, of course, our objective is to have a impact on the society. For example, like providing low cost materials to address water challenges in low and medium income communities. So it would lead to innovation such as like development of new materials, processes, or filtration system design. But it would also lead to new scientific discoveries because we are using these specialized surfaces and real world environmental conditions. The bacteria might evolve and they exhibit different behaviors or gene expressions, which we can actually learn and actually use those insights for other applications as well. For example, in healthcare, like how to prevent these hospital infections, producing like using biotechnologies for like food production or other types of water treatment applications. So this work can also lead to newer scientific discoveries. So now I would like to acknowledge our team, like our collaborators at University of Toronto. Like the work I showed in this work, like the previous slides on the nanochemistry, they are actually developed by Professor Jeffrey Ozin's group at the University of Toronto. We are further developing that and like tailoring it for bacterial applications at Imperial. And we also regularly correspond with the Global Humanitarian Development Foundation and the government and the engineers at the government of Rwanda, where they educate us what could be the economic limitations or implementation limitations, or what kind of water cleaning processes strategies we should be targeting at, depending on the resources they have access to there. And this work is funded by like Research England Grand Challenges Fund, which is like agency under the UKRI. These are the references. And to end this presentation, so the main requirement is like we are trying to meet the Horizon 2050 goals, which requires design of materials, processes, or systems that are circular, replenishable, and affordable for global communities. Thank you so much for joining this call.
Hello, thank you very much for your presentation. That was really great. If anyone has any questions, please make sure that you put it in the Q&A function now, and we'll just have a look and see if there are any questions just yet. So there is one asking where I can find the briefing papers. I can answer this one. So I've put a link in the chat to all of the briefing papers that we have available. If you want, I can just put that back into the chat again. So if anyone has any more questions specific to the presentation, please make sure that you put them in the chat. I've just put some more links into the chat. So if there are no further questions, then if you have any questions, please do not hesitate to contact us, and I can send over the questions to Dr. Cherukupally to answer them via email. So for now, I would just like to say thank you very much, Pavani, for such an informative presentation. And a recording of this webinar will be put on the IMSE YouTube channel very soon. And you can find a link for this in the chat function that I just put in there. Oh, sorry, we've just got, oh, that's fine. For the chat function, the next webinar in this series will be on the 18th of June at 2 p.m. And this is on the theme of low-cost polymeric materials with antimicrobial properties. And this will be presented by Professor Darryl Williams. Next week, we also have a webinar in the Greener Plastics Future and Sustainable Materials series. And this is on Tuesday, the 16th of June at 2 p.m. And this is by Dr. Ajay Talbot-Brandt, and it's on making and breaking materials with ionic liquids, and we'll discuss designing solvents for producing more sustainable carbon fibers and for enhanced plastic recycling. Thank you very much again to Dr. Cherukupally for that webinar, and thank you very much for coming and listening to us. Thank you.